Hello and welcome to pH Biology. Today's explainer is the active site, the small pocket on an enzyme where the substrate binds. By the end of this video, you'll know what shapes the active site, why specificity matters, and exactly what heat does to it. The active site is a specific region on an enzyme where the substrate binds. It's shaped to fit one particular substrate, like a lock for a key. The active site shape is held in place by hydrogen bonds and ionic interactions in the protein's tertiary structure. Disrupt those bonds and the active site loses its shape. Three things. 1. The active site is shaped by the protein's tertiary structure. 2. Heat or extreme pH disrupts the bonds and the active site changes shape. 3. Once denatured, the substrate no longer fits. The reaction stops, often permanently. This is the heart of why enzymes have an optimum temperature. Common slip. Don't think of the active site as eating the substrate. The substrate binds in the active site. The reaction happens and the products are released. The enzyme is unchanged and ready to bind another substrate molecule. Once a million reactions per second in some cases. Quick example, amylase has an active site shaped to fit starch. Heat amylase above 60 Celsius and the bonds holding the active site break. The pocket changes shape, starch no longer fits, and the amylase stops working. That's why we keep the amylase pH practical at 35, to keep the active site intact. Right, if any of this is going to stick beyond this video, drop into phmaths.com. You'll find a free 4-week GCSE higher revision planner ready to download, plus the mastery workbooks, one for every higher topic, every answer fully worked, every common slip flagged. Simple explanations, serious results. See you next video.